Praparat, a new artificial life model, outline its design, and present two virtual creatures that emerged within this framework. Because the session time was not finalized at the time of recording, my attendance on the day is still uncertain. If I'm not present, please feel free to contact me via email. In 1994, Carl Sims proposed a system of virtual creatures that could be simulated on a computer. While this virtual creature has a very high degree of freedom in its morphology and behavior, it has limited expressiveness in reproduction, development, and individual interactions. An example of a model that can handle reproduction, development, and individual interactions in a composite way is a self-reproduction pattern of cellular automata first proposed by John von Neumann. For example, the self-reproduction pattern loop that was proposed by Langton was robust and could achieve self-reproduction even when mutations triggered by collisions with other loops occurred. However, they are highly restricted in their morphology and behavior because they represent life as a pattern of cells. In this study, a new artificial life model is proposed that can simulate reproduction, development, and individual interactions while exhibiting high expressive power for morphology and behavior. This approach represents an attempt to combine the virtual creatures proposed by Sims with the elements possessed by cellular automata. The proposed model is based on the concept of incorporating tierra and cellular automata mechanisms into a cell that moves freely in three-dimensional 3D space. Similar to Conway's game of life, the persistent patterns exhibited by these cells can be interpreted as virtual creatures. In other words, the proposed model does not employ any human-designed fitness functions, and the forms that qualify as creatures are not predefined. This framework allows for a variety of emergent structures that observers may consider as creatures. The primary objective of this study is to explain the details of this new model and to conduct a proof-of-concept demonstration showcasing its capabilities through several experimental results. In this model, the form of virtual creatures consists of spheres, which are called cells, and springs that connect them. These virtual creatures acquire their morphologies via repeated cell division, binding with the surrounding cells using springs or unbinding. The procedure is encoded in the genes possessed by each cell, which are read sequentially to represent the developmental process. The cell can copy a portion or all of its own genes during division and pass it on to a new cell. These genes are constructed from 64 types of characters. During a mutation, these characters are replaced by other characters with a certain probability at regular intervals of time. This represents mutation within the proposed model. Thus, coupled with the selection that arises from survival competition, virtual creatures should undergo various forms of evolution. In addition, each cell has a feed-forward neural network, the parameters of which are determined by the genes. This neural network takes signals from other connected cells, as well as information about other cells and light that have collided with the cell as inputs. The output of this network then becomes a signal sent to other connected cells, or determines the actions performed by the cell. For example, if the value of the unit labeled EAT in the output layer exceeds a certain threshold, the cell will eat other cells it is in contact with. At this point, the cell can only eat other cells that have fewer connections than itself. The network also determines the amount of energy to send to other connected cells and changes the natural length of the springs connecting them. In this way, the neural network enables a response to external inputs, separate from the morphology acquisition behavior derived from the genes. Virtual creatures in this model are simulated on a 3D field with elevations and depressions. The sun is located above the field and oscillates in one direction, serving as the only source of energy in the model's environment. Now, let's take a closer look at the genes of these virtual creatures. Each cell has a string as a gene and another string that determines its readout position. Considering the difference in their roles, the gene and readout position strings are referred to as the book and the bookmarker, respectively. This mechanism can be illustrated as follows. Each cell has a book that functions as a gene and a bookmarker that indicates the readout position. When the cell opens the page marked by the bookmarker in the book, it finds instructions for itself. For example, it might say, generate a cell with a radius of 0.1 and a red color. Copy pages 0 to 120 of this book, and pass them to the generated cell. This is followed by an instruction regarding the position of the bookmarker, such as move the bookmarker to page 32. When the cell moves the bookmarker according to that instruction, it finds yet another instruction, such as enter a state waiting for connection with surrounding cells and move the bookmarker to page 81. In this way, by moving the position of the bookmarker and executing one of the 4 types of instructions written there, expansion, connection, disconnection, or transition, the creature forms its shape. Shown here is a virtual creature that self-reproduces while forming a tetrahedron by having 4 cells connect with each other, and its gene. Now, let's introduce mutations by replacing characters in these genes with a certain probability at regular intervals. We will observe how the tetrahedral virtual creature evolves as a result of these mutations and natural selection. After a while, this dumbbell-shaped virtual creature was born. Let's take a closer look at the reproduction and development process of this creature. First, a single dumbbell-shaped creature performs cell division on both sides. Furthermore, from there, white cells are expanded at each end. After that, the center divides, and the cells that were just expanded disappear. Upon investigation, it was found that this disappearance of the white cells is caused by the green cells that expanded the white cells eating those white cells. It's a somewhat strange process for a cell to eat the cell it just created itself, but it is thought that these white cells have the role of a protector to prevent the green cells from being eaten by other creatures. In other words, the temporarily expanded white cells physically keep other creatures away. To confirm this, we prepared a creature by editing the genes of this dumbbell-shaped creature to omit the process of white cell generation. The dumbbell-shaped creature with the process of white cell generation omitted in this way will be called hornless dumbbellus, and the original creature will be called Hornario dumbbellus. We place these cultured creatures in the same field. Hornario dumbbellus is on the left side, and hornless dumbbellus is on the right side. Since they are hard to distinguish, we start the simulation with color coding. Initially, the numbers of both were balanced, but gradually the number of hornless dumbbellus decreased and eventually became extinct. From this result, it is considered that the white cells were indeed working as a protector to prevent being eaten by other cells. Now, let's change the seed value, and again, try to evolve the tetrahedral virtual creature. After a while, this time, a white virtual creature with a large spread was born. To make the structure easier to see, if we display only the connections between cells, we can see that it has a reticulated body like this. In order to maintain such a large body, it is necessary to efficiently distribute energy throughout the body by the neural networks of individual cells. What's shown in this graph is the number of energy transports occurring throughout the field, and the transition of the total number of connections in the field. For a while after the start of the simulation, while the number of energy transports is almost zero, the number of connections is unstable, often increasing and decreasing significantly. This is thought to be because creatures with a high number of connections have emerged due to mutations, but were unable to properly distribute energy and maintain their large bodies, causing them to go extinct repeatedly. Let's take a look at the creatures at a time when energy transport begins to rise. It appears to have a brown body with pink legs. If we display only the connections to make the relationships between cells clearer, it looks like this. Compared to the Hornario dumbbellus we saw last time, it clearly has a more complex form. However, this also goes extinct after a while. A little later, the number of energy transport surges and the number of connections stabilizes. This is thought to be because a creature capable of distributing energy throughout its body, composed of many cells, has emerged. Let's take a look at the state of the field at these 4 points in time. The moment when the number of energy transports dramatically increased. The time after the number of connections stabilized for a while. The time when the number of connections increased by 1 stage. The time after further passage of time. You can see that the network is getting larger and the color is becoming whiter over time. As an experiment, let's sample one of these cells, fix it in the center of the field, and give energy only to this fixed cell. On the left side, the connections between cells are displayed, and on the right side, the energy possessed by each cell is visualized. As time progresses, it can be seen that energy is distributed even to places far from the central cell where energy is given, and it grows in concentric circles. Now, another thing you notice when observing this reticulated virtual creature is that there are at most 3 connections extending from a single cell. Apparently, this creature seems to be detaching the 4th cell at the same time as it expands it. Let's take a look. Focus on this cell in the center. It's hard to see as is, so I've made each cell smaller for display and highlighted the cell of interest in red. Now, let's move forward in time. Here, a new cell is extending, and then it quickly detaches. If we regard these flung cells as offspring, this behavior helps to disseminate the offspring over a wide range. In conclusion, this study proposed a new model of artificial life that could handle reproduction, development, and individual interactions in a composite manner. Through the discussion of 2 virtual creatures discovered via mutation and natural selection, a proof-of-concept demonstration showcasing the model's capabilities was conducted. The reticulated virtual creature had an interesting way of reproduction, in which it detached parts of its own body and flung them far away. It also grew a body composed of many cells by efficiently sharing energy, which represents a more advanced developmental process than that of earlier tetrahedral virtual creatures. Regarding individual interactions, the dumbbell-shaped virtual creature acquired a form of protection by expanding a protective organ during division, making itself less susceptible to predation by other virtual creatures. Taken together, these results indicate that the model affords the flexibility to address reproduction, development, and individual interactions in an integrated manner. However, this model has several limitations. To begin with, there are numerous variables that must be set before running simulations. For instance, variables to be set include the possible ranges of cell radius and mass, the number of units in the neural network's hidden layer, and the mechanisms and thresholds governing the spring's natural length and energy transport between cells. In addition, there are environmental variables such as mutation parameters, terrain, and the distance of the sun. The simulations merely represent one example of the possible combinations of these variables. Future research should evaluate the impact of changes in these parameters. However, in our current setup, such evaluations require simulations on the order of 100 million steps for the 2 types of virtual creatures presented thus far to emerge. Using an Intel Xeon W1270P, 16 cores, this required approximately 2 to 3 months to complete. Consequently, with the current approach, it is very difficult to systematically study the effects of those parameters. Additionally, the 2 virtual creatures that emerged in this study were not thoroughly analyzed. For example, the specifics of the genetic book alterations that occurred during the evolutionary process, including the methodology for such analysis, are still under exploration. As a first step toward more efficient progress, we are accelerating computation by introducing further parallel processing and optimizing the number of simulated cells. Thank you for watching.
Thank you very much, Keishu. That was wonderful work. So we have 5 minutes for questions now. As I said at the beginning, you can submit questions on Discord if you like that sort of thing. And let's just see if we've got anything there. So in the evolution channel, which is the channel for this session, so it looks like there's nothing there yet, but we have some questions in the audience. Lana.
Thank you for your talk. I really love this simulation. I'm impressed that you let it run for 3 months. I was wondering, if you let it run longer, do you expect to see any kind of ecological interactions or kind of interactions between several species, something like that?
Yeah, exactly. So actually, in this simulation, so we can finally see the only one species in this field. So as you said, so I think there are two problems. The one is the simulation time, of course, and another problem is the field side. So if we want to see such the ecological system, we have to make it bigger. But I'm not sure how much the size is required to see such a system. Of course, when I increase the size, we also it takes longer calculation time, and that's why I haven't checked it. So, but in the future, I will see it. Thank you.
Any other questions? Cliff.
How do I ask this? So I guess one question, two questions. One really quick. Where is the cost, the time cost? Is it running the ANN? Is it the physics simulation?
The first is the physics simulation, and the second one is the neural network. So because every time, we have to calculate such a neural network, for each cells, it takes a long time.
Okay, and then the second question was the output of the neural network. Does that control motion, or is it just making decisions about like extending parts and like creating cells or links, and that the motion is a result of that process?
So I mean, so could you say?
So like when it produced a new cell, and then it cut that connection, that cell moved. So do the organisms move without growth, if that makes sense? Like, do they have muscles?
So they can change the natural length of that string by output the neural network.
Okay. So there's growth decisions and there's movement decisions.
Exactly, yes.
Anyone else? I've got a couple of questions. So you had an uneven terrain, so there were mountains and valleys in your environment, and also you said the sun moved over the environment. Have you noticed any adaptations specifically to those non-biological aspects of the environment?
So you mean the effect from the field? You mean?
Yeah, is there any adaptation to the sun moving across the sky, or is there any advantage of a creature being high up on a hill rather than down in a valley? I was just wondering about the evolution to the abiotic environment.
Yeah, I wanted to see that, but I haven't seen that. Yeah, exactly. Yeah. But I think in the first I have to make the field bigger, and I think the hill is a little bit small comparing to the cell size. So that I want to I have to check the many various of the field in the future.
Right. Thank you. Okay, probably time for one more question over there. You can use that microphone just up there, I think.
Hi, thank you very much. So does every cell have a different genome and a different neural network weights, or is it that each organism shares all of the cells in one organism share the same genome? So if they're connected, they're one organism.
Each cell has a different neural network and the genes, because the neural network, the weights, they come from the genes, and the genes they're a little bit different from each other because of the mutation.
Okay. So then how do you kind of define what is an organism or what is a species? I mean, when you, like with the dumbbell one, I mean, how do you know that, well, or in the larger organism, if every cell has a different genome, how can you define that that's a single species?
Okay. So actually that is a problem. So when I do this simulation, so when I see the field, even it's almost the same. It's almost the same creature. However, the color a little bit different from each other. So I'm not sure how can we separate by the species. So I'm not sure how can I define it. So I think that I should learn from the real the creature. I mean the real creature categorized the way, I think. Thank you so much. Yeah, that is I'm still the thinking problem.
Hello. So my name is Gaylinn, and I'm presenting my paper, Ramps and Ratchets, Evolving Spatial Viability Landscapes. This started off as a class project and then evolved into a full-fledged research paper with the help of these fine folks. And in evolutionary computation, we usually start from a classic characterization of what evolution is, the survival of the fittest. But the problem is, when we write algorithms that keep just the best individuals, we quickly lose the diversity that is the engine of evolutionary progress. So to compensate, we have to find clever ways to soften selection pressure and actively manage the population's diversity. But nature is not so heavy-handed. Diversity and selection pressure emerge from local interactions between individuals, populations, and their environments. So maybe there's something that we can learn from this. Natural selection cares more about viability than fitness. Any individual that's good enough to survive gets to pass on its genes, which means that genetic drift plays a much larger role. Slightly harmful mutations are allowed to persist, maintaining greater diversity and creating the potential to evolve across dips in the fitness landscape. So this pressure to evolve more complex and intelligent behaviors actually comes from the environment and how it changes over space and time. So in this experiment, we show that environments where the difficulty varies spatially can drive phenotypes to greater fitness while still maintaining the diversity needed for innovation. We also show that for the first time, we can evolve an environment to do that job for us better than one we designed by hand. To do this, we drew inspiration from the Baim et al. Megaplate experiment, a classic paper in evolutionary biology. Here's what they did. They started off with a single petri dish the size of a table coated in agar. Across that agar plate, they laid down antibiotics in stripes of increasing concentration, ranging from no antibiotics at the edges to very high concentrations in the center. At first, any antibiotic at all is toxic to the population, but gradually the bacteria evolve more and more resistance as they migrate towards the center of the plate. Our simulation has a similar design, except instead of antibiotics, we set viability thresholds, which determine the minimum fitness required to survive and reproduce at each location. On the left, you see our Baim environment, inspired by the Megaplate experiment. And this image represents a grid of 64 by 36 locations, or demes, each with its own population of 25 evolving individuals and a viability threshold, shown in shades of blue. So like the Megaplate experiment, survival is easy on the edges, but each step towards the center requires incrementally greater fitness. On the right, you see our flat control environment, where there is no spatial variation and all individuals can survive everywhere. And we wanted to ask, does the viability gradient matter here? How does it affect the evolving population? And is the Megaplate design really the best that we can do? One nice thing about this setup is it doesn't really matter what fitness function we use. In this case, we chose one called hierarchical if and only if, or HIF. But our method should work on a wide range of fitness functions, phenotypes, and genotypes. We chose HIF because it's difficult to solve, but easy to implement. It uses phenotypes that are just bit strings of length 2 to the n, in our case, 64 bits. It divides the bit string up into overlapping substrings that are powers of 2 long, and each substring is worth a number of points equal to its length, if and only if all the digits in that substring match each other. The best possible solutions are strings of all zeros or all ones, which makes this a needle in the haystack problem. There's also no preference for zeros or ones, so different parts of the bit string might evolve in different directions, making this a deceptive fitness landscape. It's also a rugged landscape, since a single bit flip can make multiple substrings break at once, leading to a large drop in fitness. When evolving bit strings, there are a few details of our algorithm that are unusual. Technically, it's a selection-elimination evolutionary algorithm with intrinsic mortality, but let's unpack that a bit. The first thing to note is the cull procedure. Instead of choosing the most fit individuals to populate the next generation, we instead eliminate any individuals that weren't fit enough to survive and reproduce. We also kill off a few extras at random in each generation, And that, I'll say more on later, has a lot to do with selection pressure. In addition to elimination, we do also have tournament selection, but it doesn't decide who gets to reproduce. Every surviving individual replaces itself with one child, and we don't do crossover for bit strings. Then the spread procedure fills any unoccupied spaces with additional children from the selected individuals, which gives them a small advantage. When filling empty space in a DEEM, we usually choose individuals from the same DEEM, but sometimes we take them from a nearby location based on a Gaussian distribution. So in this way, bit strings can move and spread their genes across the environment. So what happened? Let's start with the flat control environment. And remember, in this setting, there is no spatial structure, and all bit strings are allowed to survive everywhere. Here we see a visualization of the evolving bit string population. The environment is 64 by 36 DEEMs in size, and each DEEM is rendered as a 5 by 5 square of pixels. Each pixel shows the fitness of one bit string in shades of red. So as you can see, the population as a whole gradually increases in fitness at a fairly steady rate across the whole environment, and it's worth noting that there is no empty space here. Since the viability threshold is 0 everywhere, bit strings only die when they're struck down at random by the cull procedure, in which case they're immediately replaced by a nearby bit string, never leaving a gap. In contrast, evolving bit strings in the BAME environment looks very different. The first thing to notice is that in the first generation, there is a mass extinction event. The initial population has low fitness, so it can't survive in the middle of the environment where the viability threshold is high. But bit strings survive just fine at the edges, and they slowly evolve greater fitness, just like in the flat environment. And eventually, a bit string gets lucky enough to migrate onto the next step up and survive there. Without any competition, it produces many offspring and spreads throughout the region. When that happens, mutations continue to arise, but the stakes are higher here. Any mutation that would drop fitness below the viability threshold is fatal. So this creates a ratchet effect, driving up the average fitness. And this repeats over and over again as the population climbs each step of the ramp, until we finally see bit strings populate the very middle of the environment, achieving a perfect HIF score. So this is a classic example of range expansion, which is a very powerful factor in biological evolution, but something we very rarely see in evolutionary algorithms. And I think another useful way to look at this is that the population is actually alternating between phases of exploration and exploitation. On each step, the population experiences high genetic drift and explores the search space broadly, but periodically, fitness improvements get rewarded with carrying capacity, exploiting innovation by spawning a whole new lineage on the next step up. And the structure of the viability landscape itself is what determines the balance between explore and exploit as the population evolves. So it was exciting to reproduce the megaplate experiment, but we were wondering, is that really the best design possible? We thought that maybe evolution could do better. This part of the experiment is inspired by endosymbiosis. The human gut hosts a population of microbes inside of it, but it's a hostile environment that selects for the right kinds of microbes. And this got us thinking, why not evolve an environment that can shape the evolution of a population within? To do this, we chose to use compositional pattern-producing networks, or CPPNs, and these tend to produce natural-looking shapes with curves, lines, and smooth gradients, like these. The basic idea is that we're actually evolving a complex function that determines the viability threshold at each location, and we did that with an algorithm called neuroevolution through augmented topologies, or NEAT, optimizing for environments that evolve the most fit bit strings within. So what did we find? On the left, you see the BAME environment, just for reference, and on the right, you see the best CPPN environment that we evolved for the same hyperparameter settings. It's interesting that we still see a gradual ramp up in difficulty, like in the BAME design, but instead of one broad ramp up and down across the whole environment, we see many tiny ramps offset from one another. When we evolve bit strings here, we see reservoirs with a low viability threshold making diagonal stripes throughout the environment, And the populations there gradually evolve greater fitness. They start climbing all the little ramps in parallel all over the map. And eventually, one bit string achieves a perfect score and is able to spread across the whole environment. One explanation for why this design works is that maybe having lots of little ramps with a gap between them is creating many parallel evolutionary searches segregated so that it's harder for one innovation to sweep the whole environment and crowd out the other possible solutions that are still evolving. To get a little bit more quantitative view on these results, here's a chart showing the mean and max fitness for evolving bit strings in all 3 environments. And this is the result of 100 replications with the outliers from the top and bottom 15% omitted. So the most notable thing here is that both of the environments with spatial viability perform much better than the flat environment across all of the replications. But we can also see that the CPPN environment achieves higher scores overall than the BAME environment. If we do keep the outliers, then we see more overlap between the BAME and CPPN environments. In both cases, we can actually evolve bit strings that get perfect HIF scores, but happens 4 times more often for the CPPN environments that we evolved compared to the BAME environment. So just one important caveat to all of this. The results I've shown you so far come from a setting where the explicit selection pressure was unusually low for an evolutionary algorithm. And things look different when we turn the pressure up. So in this paradigm, there are 2 key factors that determine selection pressure. Mortality determines the rate of turnover in the population. And without this, every viable bit string in a DEEM would have 1 child, and then additional fitness wouldn't actually confer any benefit. So by randomly killing off a few extras, we make space for those extra children to be populated. The other factor is terminant size, which determines how much fitness influences selection. If tournament size is 1, then we choose bit strings at random to make those extra children. But if it's 25, then we always choose the most fit bit string in a DEEM. So what happens when we change these settings? Well, we did a hyperparameter sweep, and what you see here is a heat map comparing 2 different experiment conditions. Each pixel corresponds to 1 unique setting of mortality rate and tournament size, and the color indicates which condition did better in that setting. Here we're comparing the BAME environment to the flat environment. Shades of blue show where the BAME environment did better. Yellows show where they did about the same. And reds indicate where the flat environment did better. The BAME environment has a big advantage when selection pressure is low, whether that's because of tournament size or mortality rate. But when selection pressure is higher, it tends to do a bit worse than the flat environment. So what this shows is you don't actually need spatial structure or viability constraints to solve HIF, but you also don't need explicit selection pressure, which I thought was very surprising. The viability gradient itself can induce selection pressure when it was otherwise absent. Next, we evolved a CPPN environment for each hyperparameter setting and compared it to the best control for that setting. And the story here is overwhelming neutrality. The evolved environments could induce selection pressure where it was absent, but they didn't get in the way when the selection pressure was already strong. Using the environment to generate selection pressure is cool, but it's not needed to solve HIF. So what is this good for? Well, for one thing, it's not always easy to compare the fitness of 2 individuals, especially in ALIFE projects. So using viability can help, since you can use a simple test or barrier for selection and not worry about who did better, just so long as they passed. For another thing, sometimes it's useful to keep selection pressure low, just to find as many diverse solutions as possible. And that is because sometimes fitness functions are complex, right? So maybe you want to optimize many constraints at once, or maybe you want to find all the possible solutions to a problem. And prior work shows that spatial environments are actually great for this, but designing them by hand can be difficult. Why not evolve them? We showed that it's possible. Lastly, diversity maintenance can help a lot with simulated evolution. For example, MapElites is a popular algorithm that keeps an archive of diverse solutions that keep evolving. But a person has to decide what kinds of diversity are worth preserving. But we think that a spatial viability landscape could play the same role with less human bias. So thanks. Do check out the source code for this project on Codeberg. Come talk to me or drop me an email if you have any questions.
Thanks very much, Nate. Very interesting. Any questions? Emily?
Really cool work. I'm curious, how do you think the results would be different if instead of having these spatial locations with different mortality cutoffs, you instead had just kind of different buckets in your population? You had a well-mixed population, but like to get into the second bucket and have access to that extra population size, you needed to have a certain fitness.
Yeah, that's a great question. You know, the design that we came up with in the evolved design had all of these little independent ramps, right? And that makes me wonder if actually what it's showing us is that you want a ramp of size 4 and you want a parallel implementation of them. So I don't know if that's just the answer to this, and there's a simple design that we found by evolution. But my suspicion is, at least for a more complex fitness function, it might be interesting to have other shapes. And so I'd be really curious to try that. Like, for instance, I could imagine evolving high skill in 2 different traits and then somehow bringing those populations together to mix. I was not able to see that with this design, but I think that's quite possible.
Any other questions? One more here.
Hello, Nate. Thank you for an amazing talk. My name is Klaus. One clarifying question about the fitness. Is all zeros and all ones equally best fitness? And as a consequence of that, did you try functions with more possible maximum fitness? And if you observe, because that's one thing that I think it would be really cool to see different areas colonizing different optimal peaks.
I'm not sure I understand the second part of your question. The first part, yes, all zeros and all ones are exactly the same.
Okay.
It's just that they match each other. But could you clarify the...
I think we can do that in the conversation later.
Happy to talk later.
Thank you.
No? So one observation of what might be happening is a founder effect. So at low selection strength, even if somebody at that boundary, so if anytime that a mutant shows up with slightly higher fitness, it isn't sufficiently higher to make a difference, then selection can't tell the difference. And so it may establish, right? So I'm assuming that, like, maybe, did you have any replicates on flat where they achieved high fitness?
No.
No,
none of
okay,
them.
so no, okay.
Yeah.
So the establishment rate may just be very low, but if you had made the world very, very large, then maybe by chance someone would establish. But then what would happen is whoever was the first to make it across the boundary would establish with the higher fitness. And then when you had the very, very small ramps, you have a lot of potential, right? So it wouldn't be one establishing. Each one becomes its own little establishing deem. And so the probability of getting an extra high one would be higher. But
Yes.
maybe that's what we were seeing.
Yeah, I think that's very close to my own analysis of the situation. And in fact, in the biology literature, there's this notion of the serial founder effect in range expansion phenomena. It's literally describing this thing. Yes, I agree.
Okay, time for one last question if there are any. Lana.
Thank you. I was wondering why the white guys didn't invade the whole space. Is there a cost to being the fittest?
That's an interesting question. I'm not completely sure. I suspect it has to do with just how crowded the low threshold areas were, right? Maybe if the mortality rate was higher, you'd see the perfect scores spreading more evenly throughout the map. But there's interesting examples from biology where you see in similar range expansion phenomena, you see a gradient of phenotypes just because it's harder for things to remix and move further back. But maybe if I let it run long enough, it might become more homogeneous. I'm not sure.
Okay, thank you very much, Nate. Our next speaker is Victor, Maull, and Miquel. Victor, do you want to get...
Well, hi everybody. Thanks to ALIFE community to host me. I'm Victor. I'm based in Barcelona in the lab of Ricard Soler. And I titled my talk as Mapping the Road to Synthetic Ecosystems. And the whole presentation goes around a central question, which is can ecosystems be designed? This, of course, is a very difficult question. That's why the effort that we did is try to map the road towards them, like both experimentally and theoretically. And by synthetic ecosystems, we mean like actually intervening, usually with synthetic biology, natural ecosystems that they are often like degraded or experiencing heavy pressures, and maybe could we like take them back from the brink and restore them. No? This kind of intervening in nature. How to get there, right? But before going to the topic, let me guide you through an example that I think is very interesting and I think you will like. I need you to go through to an island in the middle of the Atlantic Ocean. This point here is the island of Santa Elena. But this is just to situate you where Napoleon died. But where I want to go is to this heart. This is farther west, and it's the island of Ascension, and it's one of the most remote places on Earth. This island looks like this, like an arrow shape. It's part of the English overseas territory now, and the most visual, the first thing you see when you look the island from the top is this green patch around the summit. This, in fact, is a cloud forest, very humid and very biodiverse. And if you go down to the ground, you would see that you can find a very weird mix of species. You can find a forest of bamboos, together with thick canopies of ficus trees, also Norfolk pines in the creeks or in the top parts of the mountain, and also when you go to the coast, fig trees and shrubs appear, right? This, in fact, is super weird, this mix of species, but this is not the weirdest. The weirdest is that this place was not always like this. And what I mean by that? What I mean is that this island, to give you an historical perspective of this place, was discovered in 1501 by the Portuguese. And at that time, it was described as a place without water, without green, just a rock, a place to anchor. There's the first visual record of this place was in the 17th century by, this is a nautical chart by Dutch sailors, drawn by Dutch sailors. This is the island, how it looked for them. And there's a small poem that allows you to see how difficult was this place. It says, "No land fertile or rich in drinks belongs to me, yet the shore is rich in fish and fowl. Completely furious in red, I rise amid the waves, where the torrid zone sees the southern pole." As you can see, it's like a very heavy place, very extreme. There's another example. I will go for it very quickly, but this is in the 18th century. They found like a passing boat, English. They found a diary of a castaway that was left there as a punishment, and they published the diary in England. You can read it. It's like an amazing story. And this guy survived in the island for half a year, and actually, from the ecological perspective, is like a meteorological station, telling us how the weather was every day. And he describes, like, again, no water, it's never raining, nothing green. Probably the whole island was looking like this. This is part of the coast that's still arid. But this is to give you a view of how this place was, right? So what happened here? How was this transition? This transition began because at the early 19th century, the English Admiralty built a base there, and they wanted to make this base self-sustainable. And actually, Darwin is involved in this story because when he was coming back from his trip, he stopped at the island, since it was a common stop from English ships. And he had the idea of greening it. And he spoke with Hooker, Joseph Hooker, at that time was the director of the Kew Gardens in London, and they decided to go for a huge plan of planting, and they brought thousands and thousands of plants and seeds to the island from 1843 to 1870. And in this short span, they created the ecosystem that we see now. No? So this is a transition from desert to green, like crazy, and the first in history. And this place allows us to ask very interesting questions about ecosystems and how they create, starting from the fact that this ecosystem was not created in evolutionary scale, it was created in an ecological scale. So questions that this system allows us to ask is what constitutes a functional ecosystem, or which processes allow the transition from bedrock to a large ecosystem, or is the ecosystem I observe at a given moment the one with the highest biological value? Or can ecosystems be designed or engineered? If so, by what criteria, at what scales, and with what degree of control? And the last one, if ecosystems can be truly designed, what are the qualitative and quantitative steps toward doing so? Of course, those are many questions, difficult answers of all of them. But a way to start is to create a map, no? This qualitative and quantitative map, what to do experimentally and theoretically. We believe that in the crossroads of synthetic biology and ecology, there's a good chance to give answer to some of those questions. And we conceptualize this road as a multi-scale problem, right? That basically is constituted by 3 scales. At the lower part, we have the microbial microcosms, are ecosystems very controlled that you can do in the lab with relatively simple theory connected to them. The next step would be closed ecosystems, closed in matter but open in energy with their theory. And the final step would be the biosphere itself, right? This vision is not new. This was a philosophy started by Odum, like a great ecologist of the 20th century. And, for example, is like well-reviewed in this book. To map this multi-scale problem, we used the concept of the morphospace. A morphospace is a very useful method to analyze a system. For example, here you need to, a morphospace is of course a 3-dimensional space that by defining 3 axes that represent the main variables that define this system according to your criteria. And when you have this space defined, you can put qualitatively examples that are at the edges and also that complement, or see that fill this escape. By doing so, you can explore the space of the possible. No? For example, here, to situate you, Ascension Island is this blue dot. In the corner is maximum succession. Succession means how the ecosystem changes over time. Species diversity, how many species are in the ecosystem. Anthropogenic driving, how much anthropogenic pressure the ecosystem have. At the lower part, this orange dot is the single species chemostat, which is like maximum anthropogenic driving because it's a laboratory experiment. Species diversity, 1, succession, 0. No? And the green dot is the undisturbed ecosystem. This is a dot. It might sound rare, but it's not rare. In fact, the rare is that an ecosystem is undisturbed. All ecosystems in Earth nowadays experience, to some degree, anthropogenic driving. Actually, they are described as novel ecosystems. You can think about them like human influence through abiotic, biotic, and social components and their interactions creates ecosystems that differ from historical ones. Many of these systems self-organize and develop novel qualities, even without continued human management. No? Those systems lie in this gray area that extrudes up, and you can find there like rangelands, gardens. You can find sewage. You can find the urban microbiome. You can find even the plastisphere, no? This accumulation of plastic in the ocean that is creating new communities novel to life. No? Once you increase the anthropogenic driving, there's a boundary that that defines wheel of design. And this boundary, this area here in yellow represents these synthetic ecosystems, right? Usually designed using synthetic biology within ecological constraints. This area will also strut up, no? As I was saying before, this problem is a multi-scale one, and of course, every scale has its own position in this cube as nested cubes. The tiniest cube inside represents the synthetic microcosms, all these lab experiments. The inner middle cube, C2, represents closed ecosystems, and the big one, the bioengineered ecosystems in the open. So the first one, this tiny cube in the middle. What can we do there? No? For example, this is a great space to experiment with the easiest configurations you can think about, ecologically speaking. And it's a place that you can ask, for example, the basic relationships in ecology, like cooperation, competition, parasitism, predation, by doing very simple experiments and applying like relatively easy theory. No? For example, the first example here is competition by using a petri dish and a couple of species, one glowing in blue and the other glowing in yellow. They start growing from the middle and they compete for resources and space. And at the perimeter of this circle, you find competitive exclusion. No? And this is, and if you do that like in a well-mixed scenario, this can be very well mapped or very well predicted by simple Lotka-Volterra equations, right? Another example of that, in this category, could be doing cooperation by introducing auxotrophies to a pair of cooperating microbial species too, and the same result it can be obtained, right? By using simple equations, you can arrive to these attractors. This would occupy this space. Of course, you can find more examples there and can be discussed, but this is the idea of this level. But in general, these systems usually lack environmental constraints, right? And this fact can be tackled at the upper level. The upper level is closed ecosystems. As I said, they represent ecosystems that are closed in matter but open in energy. And historically, they started, or the first time they were like explored deeply was related to space exploration, and usually for human sustainability within closed ecosystems, usually in space or in this kind of setups. Two great examples that maybe some of you know are Biosphere in the USSR, that was a facility to sustain humans by producing plants. And also a famous example is Biosphere 2 in the USA. But other configurations like Winogradsky columns or panels that are also in this space of the closed ecosystems are lower and easier instant examples of these closed ecosystems that can be useful to ask fundamental questions in ecology in a relatively 2 minutes. Oh my God. Okay. Okay, like theory that you can do around these systems, these closed systems is this, right? Think about like the easiest way you can think about a closed ecosystem, an open ecosystem, is like you have a species that is living in a lake, consuming a resource. There are incomes and outcomes, and the resource can be recycled by these same species. If you do one step of closure, you close the income of species, and you homogenize the income of resources and the outcome of resources and species. Then you have a chemostat. If you do one step of closure and you close all the flows, you end up in this system in the lower part, and you impose logistic control by space. If you solve this system, you end up in this phase space that tells you that the recycling rate needs to be greater of the inverse of the growth rate to life to exist. This is a super restricting scenario, right? And in this setup, it can be useful to ask questions like, can biodiversity in this setup relax this condition? If that, how, right? And finally, we end up in the bigger cube, right? And this bigger cube builds up usually looking and mirroring in novel ecosystems, and many of the times introducing synthetic species in these ecosystems to restore them or to bring them to different scenarios. Here, they are like remediated toxic landfills, synthetic plastisphere, or drylands, which are great examples. Theory of that, of course, experiments in this section cannot be done if you don't do experiments in the other cubes. But some theory that we did in our lab tackle fundamental questions at this level. And well, this has been a very short summary of this paper that you can find in ACS Synthetic Biology. Thanks to my team, and thanks all of you for listening, and I'm very up for questions. Thank you.
Thank you very much, Victor. Any questions? Emily.
Really cool stuff. So I'm curious, on the axis you had going up, the succession/development axis, in your work, how much are you thinking about evolution also being a part of that, like maybe at the microbial level, versus it just being pure succession?
Yeah, in my work, I mean, what I did until now, we are based a lot on the ecological scale, because we believe is a fundamental first step. Like, for example, the example that I was talking at the beginning, one ascension, proves that the ecological scale is very important to make some robust predictions, but of course the evolution is the pending subject, and it's the most difficult to tackle also, but absolutely.
There was another question here, I think. Yep.
Yeah, very, very nice talk. I was wondering if you try to assemble or design an ecosystem which is at the systems level. How do you deal with the problem that if you would, for example, modify or breed or select organisms, that they work on this different level? So if you want to like genetically engineer bacteria, how can you engineer it that it works well with different organisms or like in assemblies that you might not even have thought of when you want to deploy them?
Like, you mean like if you are thinking in the lower level and you are designing something that works there,
yeah,
how can you be sure that this will work in the upper scale, right?
Yeah, or how can you...
Yeah, absolutely. That's why it's super tricky how synthetic ecosystems might work in the upper scale. Both if it works is a difficult question. Usually synthetic biology dies in the open. And the other question is, if it's not dying, how to be sure that what you are introducing in this ecosystem is doing what you want, no? Both control and prevention. To do that, there's several aspects, mechanisms that you can use. For example, this is something that we, for example, in this first paper, we were thinking on how to use biodiversity as a firewall to control species that, for example, you can take a species that is living already in the system, modify it, and bringing it back. Then this species that comes new to the ecosystem is really not new. It shares all the connections with the species that was already present. This is one way. But of course, you need to go to the lab first, no? This step might be tricky. So there are other mechanisms, like, for example, you can think about using horizontal gene transfer. This is something you can engineer in the lab, like at the level of a plasmid. And you can use it you can introduce it in natural communities and have some control of how this mechanism is spreading. So you don't need to pass through the laboratory level to engineer the species. Those are ways, but it's...
Okay, thank you. Mm-hmm.
Okay, I think we should get on to the next talk. So thank you very much again, Victor.
Great.
Okay, so our final talk for the day is an online talk. Just to remind those who weren't here at the beginning, this was originally scheduled to be the first talk of this session. It's Niels Fontijne, who will be talking about towards biologically plausible encodings in the evolution of artificial creatures.
We will discuss the introduction, the methodology, the results, and the conclusions. Our work is located in the field of evolutionary robotics, a subfield that evolves autonomous robots by means of artificial evolution. One of the topics that has been studied is the efficiency of natural development. During natural development, chemical gradients define the body axis, and the chemical gradients guide temporal unfolding and local interactions. As an example, we have a hypothetical embryo with an anterior-posterior gradient along which the body develops. We also have a symmetrical left-right gradient that guides the organism to develop similar legs and wings on both sides of the body. These studies have led to multiple artificial developmental encodings. We will list some of them, ranging from a high abstraction level to a low abstraction level. For a high abstraction level, we have a CPPN. Then we have a medium abstraction level L system and a lower abstraction level TRN. Veenstra et al. Compared a CPPN and an L system against a direct encoding for the co-evolution of robot bodies and brains. Generally, the diversity levels increased when the abstraction level was lowered. However, morphological convergence occurred earlier for the CPPN and also earlier but a little bit later for the L system. Similarly, final task performance was lower for the CPPN and equal for the L system. However, we have no idea how this will be for lower abstraction level encodings like the TRN. Therefore, we will explore the effects of abstraction level on the produced artificial creatures, and we will identify and discuss their biological plausibility and address one of the issues and evaluate the resulting encoding together with the other encodings. We hypothesize that more biologically plausible encodings exhibit higher diversity levels, and higher diversity levels are important when we want to deploy robots in unpredictable environments or if we want to change the task of the robot. Now we move on to the methodology. In our research, the robot is modular and consists of a core brick in red, unmovable parts in blue, which are called bricks, and hinges in white, which facilitate movement. The movement is guided by central pattern generators. To be able to evolve the robots, we need representations for the brain and the body. First, we have the representation for the brain, which is given by a CPPN. So we feed a certain coordinate to the CPPN, and the CPPN outputs the oscillator weights. For the body, we have three kinds of encodings. The first one is the CPPN. We feed a certain coordinate to the CPPN, and the CPPN outputs a certain type and rotation of module. Further, we have the GRN based on artificial ontogeny. We have a sequence of numbers which represent a gene, and the gene can be expressed, which initiates a continuous loop of production, diffusion, and decay of transcription factors. The concentrations of the transcription factors decide on cell placement and which module to place. In our paper, we mention multiple biological plausibility issues of the GRN. Here we will list some of them. First, the diffusion process seems not to displace any or almost any transcription factors within and between cells. Further, we noticed that the gradients will always be unstable, like the dashed line, where we would expect something like the thick line. This is especially worrying as the gradients are hypothesized to drive the efficiency of natural development. We also noticed that the modeled processes are non-concentration dependent and non-parallel within cells and across cells. An example of non-concentration dependency is visible in this image, where on the left side we see a concentration of 0.8 and we have a diffusion rate of 1. So we will end up negative with minus 0.2 and we have an overshoot on the other end of 1.2. As an example of the non-parallel processing of the GRN, we here have an illustration of how the GRN models the processes. So in black we have the cell sites and the surrounding boxes are the cells. The GRN starts at a certain cell site, goes to the next cell site, then the next cell site, then the next cell site, before it goes to the next cell and repeats the process. As a solution, we propose the more realistic GRN, which models for every cell site the decay, the diffusion, and the production. The decay and the diffusion are both concentration dependent. The formulas are combined in matrices. The use of matrices implies that the cells are processed in parallel. And we also have a more stable estimate over time as we apply the Craig-Nicholson method by evaluating both a t and t plus 1. To illustrate the effects on the computed gradients, we have some GIFs. On the left side for the GRN and on the right side for the more realistic GRN. In the GIFs, a red color indicates a higher concentration. So here we have for the GRN, you clearly see all cell sites. And on the right side, we see for the more realistic GRN. If we look closer, we see that the concentrations in the GRN are limited to a single cell site, and they don't spread out to other cell sites. If we look at the more realistic GRN, we see that the concentration flows out to the neighboring cell sites, and that the gradient is really gradual. So we have red on top, then light blue, and then even lighter blue, as would be expected. We evaluate the encodings as following. For every encoding, we perform 20 trials, where we perform random search for 50 generations to measure the biases without applying selection pressure, and we perform evolution for 600 generations, where we optimize for speed in the x direction on a flat terrain. We evaluate the robots for 30 seconds in the robot framework, Miquel and Revolve. Now we move on to the results for random search. First, we evaluate the sampling rates on the y-axis and the unique robots on the x-axis. We see on the left side that the GRN-based encodings seem to have a higher bias for the most prominent robots. On the other side, we see that the GRN versions seem to develop more unique robots. When we zoom in to the most prominent robots, we see that the GRN versions produce robots that are most similar to the robots that would be expected based on random sampling, which is shown on top. The CBPN produces completely different robots. We can also show this by putting the number of bricks on the y-axis and the number of hinges on the x-axis, and coloring the density from low in blue to high in red. We see that the CBPN mainly samples from the diagonal, while the GRN versions are able to sample a wider part of the space. Especially the left bottom corner, which indicates smaller robots. And if we look at the random sampling, it's more likely that you get a higher density, so a higher bias for the smaller robots. Besides sampling frequencies, we also measured some morphological descriptors, such as the number of attachments to the core. In blue, we have the density of all robots, and in orange, the densities of the unique robots. Based on such plots, we saw that lowering the encoding level leads to more diversity in joints, limbs, and lengths of limbs, and also more asymmetry and variation in symmetry. Now we move on to the results of evolution. First, we didn't see any significant differences in final task performance between the encodings, although we saw that the CBPN shows significantly faster evolutionary progress. We also notice that the less abstracted the encoding becomes, the more safe the behavior becomes. On the left side, we have an example of safe behavior. The organism rolls forward. And on the right side, we have an example of unsafe behavior, where the organism just rolls over all the time. Besides behavior, we also measured the morphological convergence over the generations. On the left, you see this for the relative number of modules, and on the right for the attachments to the core. And in general, we see that the lower the abstraction level becomes, the later the convergence occurs. We also measured diversity. First, for the population level, we did this by the summed Euclidean distance between the members of a population. And on the left, you see this for the relative number of modules, and on the right, summed over all morphological descriptors. And in general, we see that the CBPN shows lower diversity than the GRN-based encodings, and the lower the abstraction level becomes, the later the diversity starts to decrease. Further, we assessed the diversity at the elitist level. Here you see the building plan for the best performance of every generation, labeled from old in yellow to new in blue. And you clearly see that there's more variation in building plan for the GRN versions compared to the CBPN. And we also categorized the best performing robots at the end of evolution for all encodings. And you clearly see that there is a more balanced distribution for the GRN versions compared to the CBPN. The bias of the CBPN to the cross-quadrupeds is certainly not due to fitness, as on the left we see that the fitness of those creatures is generally lower than those of the snakes and the two-limb creatures, which are displayed on the right. Move on to the conclusions. We see that lowering the abstraction level of the encoding does not lead to any performance gain, and we even see slower evolutionary progress. However, we also see higher diversity levels, slower morphological convergence, more safe and intelligible behavior. Maintaining higher diversity levels for longer is especially helpful in new unpredictable environments, where we certainly don't want to run out of diversity. However, our results first need to be confirmed for different tasks and environments. Here are our references, and thank you for listening to our presentation.
Well, thank you very much, Niels. I don't think he can hear us, so I want to apologize to him for the technical difficulties. So we won't be able to do questions and answers with him just now, but his paper is, of course, in the conference proceedings, and his contact details are there, and I'm sure he'll be very happy to answer any questions by email. And that brings us to the end of our Evolution 1 session. Despite all the problems, we're actually on time just now, so fantastic. And I believe the Evolution 2 session is due to follow on right away. So thank you very much to all the speakers once again.